Good afternoon, everybody. Now we go back to the Mediterranean Sea after Banyuls, which is a little bit farther. And we are in the Gulf of Naples with our time series. It is based at the station MareChiara, that is now called LTER MareChiara because it is part of this long-term ecological research program, international, European, Italian. It is a nested network. The aim of our time series is understanding the diversity, evolution, and functional aspects of plankton organisms in relation with environmental conditions, mainly. And the series goes on since 1984. It has had some interruptions, though. Well, the main and the only institution running the time series is the Stazione Zoologica Anton Dohrn, which is one of the oldest marine research institutes in the world, founded in 1872. And there are 3 labs involved in this research with 8 researchers and 3 technicians. Obviously, this is not for none of them is the main research. It's something that we carry on since many years as a side research program, not the main one. And our facilities are 2 research vessels. One of you can see it on the right down bottom corner, and this is the one that samples every week. And we have a molecular biology service with the capillary sequencer, bioanalyzer, robot, and there are some next generation sequencer, one that will be purchased this year. We have equipment for environmental analysis, meaning nutrients, particulate organic carbon, and so on and so forth. Taxonomic expertise for phytoplankton, zooplankton, and zoobenthos, but this expertise is not based on our time series. There is another time series close by in the island close by. And then we have scanning and transmission electron microscope. I say Stazione Zoologica is the only partner, but obviously it takes advantage of networks of excellence. S3, to which it participates, actually is the leader institute in MBRC, is participating in LifeWatch, is part of the consortium Euromarine, is in the project Assemble, that are all these projects that allow to collaborate among different institutions. And also, I have already mentioned belonging to the long-term ecological research program. And there have been several programs, national and international, over the years, through which we are supported for part of our activities at the station. And also, for the data analysis, we are linked to this SCORE, Scientific Committee on Ocean Research, concerning time series analysis of plankton. Well, the contextual data, I very much support the presentation of the colleagues from Plymouth. Yes, these are our main data. And the main data would be phytoplankton, mesozooplankton, microzooplankton, and the contextual data for us are the environmental data with which we compare our data. And where's the molecular data? Yeah, we started some years ago with molecular data, starting mainly with phylogenetic markers for taxonomy purposes. We wanted to know actually what were the species that were there. So most of the data are based on DNA taken from cultured specimens. So it's I mean, not metadata. And we have this data concerning several phylogenetic markers from 18S to much more variable ones, such as microsatellites for individual species. And then instead, speaking about clone libraries, we have few clone libraries from 2005 and some generated with the genus-specific markers on some specific genera. Then we have a few dates with 454 sequences coming from European project Biomarks, but we are planning more for next year. Microarrays for harmful species. And then we were visited from the Craig Venter Institution cruise a couple of years ago and participate in the Ocean Sampling Day. We have sent some samples from our station for transcriptome sequencing to the Moore Foundation, and we have these frozen filters. Accessibility, yeah, we have database under construction. MASISTE and Europe are all projects which help us, and the data are available on request. We are working on a data policy. We have no plan for molecular data. They are scattered, really. They are in the worst condition, I would say. We will have a website for our station in 1 year, and of course the sampling site is available if you want to come through Assemble or also with other means. Thank you very much.